Hi, this is Dr. Mead. I'm going to talk to you quickly about the open response. Sorry, I've had a little technical trouble getting everything online a little later than I thought. Um, and I'm using a free version of this one, so I only have 15 minutes, but that's good. I'll keep it short. So this was the first question. Protein synthesis is vital for cell growth and metabolism. So make sure you identify the four parts when you write an open response, separate out A, B, C, and D, but then write in complete sentences. I recommend you take notes to make sure you know what you're talking about and you don't just babble on, but if you don't have time, you don't have time. So describe transcription and translation. Um, Given that they're looking for more specifics later, I would keep this part really simple and say that transcription is, taking DNA and writing messenger RNA from it. And I would use some details of that process, a couple of the enzymes, um, and likewise for translation. Not every single detail because you're going to say more in the next bit, but make sure that you add enough to make it sound like you know what you're talking about. Um, so it might be an advantage for outlining for this one just so that you don't repeat yourself. Similarities between transcription and translation and differences. Now, I think this is kind of a weird question because they're two so different processes, but this came from 2005 AP exam, and the link here um, will take you to those. You can't click on it in this movie, so you just have to copy it, or I'll include it in the email that I send you with the link for the videos. So similarities and differences that you would come up with is, similarities is that they're both a part of taking a gene and creating the phenotypic expression of a gene. Um, Differences are that, one takes place in the nucleus, one takes place at the ribosome. Um, one works directly with the DNA inside the nucleus. The other is a copy. You might want to say that the DNA is, um, the entire DNA the same in every cell in a body, but the segment of messenger RNA that's being transcribed is only the part that's necessary to make the protein. And then lastly, the structural changes that can occur to a protein after translation to make it function properly. So there we're talking about going from the primary level of a protein, amino acids linked by peptide bonds, into being folded and twisted and made into the shape that it's supposed to be. So the four levels of structure of a protein. And I know for some of you that's been like a nightmare. Every time it comes up, you say, "Oh, I don't know it." So this would be a great time um, to sit with that chapter, I don't know, 3 or whatever, the page that shows you the four levels of a protein. Okay, so, oops. Oh, there it is. Um, Viruses are considered non-living and bacteria is considered living. Make and defend the idea that viruses are living, or make and defend the case that viruses should not be considered living. If you look at the PowerPoint on Schoology that we went over for viruses, there's a nice chart there. This is something that's still being argued. The main argument about viruses as being living is that they have genetic material. Um, they pass on genetic material. They have certain behaviors. Gosh, I can't think of everything off that list right now. But in the case for non-living is that they are basically parasites, that they don't live and reproduce without a host. They don't necessarily respond to their environment. But back to the fact that they are living, they do evolve. So that's a huge point in the living side. Heredity at the molecular and phenotypic levels is complex and necessary for species survival. Name and explain one type of prokaryotic gene control. Well, there I'm talking about the operons. So you would pick one of the two operons and explain how that's part of prokaryotic gene control. So in the case of tryptophan, um, it's always on unless we have enough tryptophan and then it turns off. Okay, so we're controlling the gene by turning it on or off. For eukaryotic gene control, you could talk about acetylation or methylation of DNA. So again, kind of promoting the expression of a gene to be on or off. Lastly, mutations. And I wanted to make sure, since that exam was a little bit incomplete just based on where I was at the time when I made the practice exam, that there really weren't a lot of questions about mutations on there, but you can expect for sure questions about mutations, and that was part of why I included this question for the open response. And here we're talking about more molecular level, whereas the last test we were talking kind of globally about chromosome abnormalities. So the mutations at the molecular level, page 328, the types of point mutations are the ones that I am looking for you to be familiar with. This is part of chapter 17. So missense, nonsense, base pair substitutions, insertions, deletions, mutagens. So I would put those on some flashcards to just quickly kind of commit those to memory so that you could explain one. So this question, heredity question here, is all about kind of turning genes on and off appropriately, and what happens when things go wrong. So this in chapter 17, this whole chapter is called From Gene to Protein. So think about it in the fact that making the wrong protein is making a change in the phenotype. So a change may not have a big effect, but some changes can have a huge effect in the organism. So, and we kind of will come back to that when we talk about the systems of the body. So this one's kind of short and sweet. I hope that helps you kind of think about it. Remember what that science teacher said in the TED Talk, that you want to keep coming back to being able to explain things at kind of a big and basic level and then feed in the details. I think that will help a lot of you stay a little bit more clear about what you're doing. All right, good luck.